Hello to everyone who is watching. My name is Caio César and I'll be presenting in this video the article titled Sanitary Quality of Soybean Seeds Treated with Fungicides and Insecticides Before and After Storage. In order to achieve high soybean yields, it's necessary to use high quality seeds associated with management practices such as seed treatment. In some cases, seed lots with high vigor may be 9% higher than with low vigor. In the production of soybean seeds, the harvest does not happen in the ideal time for the planting of the next harvest, which requires a long storage period of the seeds until sowing. In this way, storage is a step in which the rate and intensity of the deterioration process should be reduced in order to maintain the initial quality of the lots. Therefore, the durability of seeds is influenced both by the initial quality of the lots as well by the storage conditions. Nevertheless, ideal storage conditions may favor the survival of several storage pathogens, including fungi from the genus Aspergillus, Penicillium, and Fusarium. In this context, it's necessary to use seed treatment, a process that consists of applying compounds capable of protecting seeds against deleterious effects caused by pathogens, aiding to control storage fungi and protecting the seedlings in the initial establishment period of the crop, favoring sowing, emergence, and development. But certain limitations are worrying, including the possible effects of active ingredients on seed quality during storage and in the field, such as reduced germination rate and emergence of seedlings due to phytotoxicity. Thereby, there is a need for studies evaluating the effect of the most commonly used seed treatment products and the correct application time on the sanitary quality and the physiological performance of soybean seeds. The aim of the present study was to verify the effect of fungicide and insecticide treatment on sanitary quality and physiological performance of soybean seeds before and after storage. The seeds used to perform the experiment were supplied by the company Nideira Sementes. Seeds from the cultivars NS7494, NS8693, and NS7338 iPro were used, which were analyzed separately by a completely randomized design in a 3x6 factorial design. Each cultivar was subject to 3 treatments: treated and evaluated, where seeds were evaluated immediately after chemical treatment; treated, stored, and evaluated, in which the seeds were stored with chemical treatment for 2 months and then evaluated; stored, treated, and evaluated. The seeds were stored without chemical treatment for 2 months and then were treated and evaluated immediately. The cultivars were also subjected to 6 mixtures of fungicides plus insecticide. In order to apply the mixture, the products were previously mixed in a petri dish and then placed in plastic bags with 2 kg capacity, being finally added 500 grams of seeds. It was all stirred until an homogeneous mixture of seeds. After the treatment, the seeds were arranged in the shade at a temperature of approximately 25 degrees Celsius for 20 minutes in order to dry the product on the seed surface. For the storage procedure, the treated seeds were conditioned on corrugated fiberboard packages and kept for 2 months in a conventional warehouse under uncontrolled conditions, with an average temperature of 20.4 degrees Celsius and a relative humidity of 65.5%. The healthy test was performed to evaluate the presence of pathogens. 8 subsamples of 25 seeds were distributed in 15 cm diameter petri dishes containing 3 filter paper sheets previously sterilized and moistened with sterilized water, agar, and 2,4-D. The plates were incubated at 20 degrees Celsius and 12 hours photoperiod, where they remained for 7 days, being then evaluated in relation to the presence of pathogens. The incidence was evaluated as percentage of found fungi. As for the germination test, 4 replicates of 50 seeds per lot were used and seeded into gemitest sheets of paper and covered by one sheet, moistened with distilled water in an amount equivalent to 0.5 times the weight of the dry paper. The seedlings were kept in a germinator at 25 degrees Celsius, and the evaluations were conducted at 5 and 8 days after sowing, being the results expressed as percentage of normal seedlings. In the cold test, 4 subsamples of 50 seeds were used. The sowing was performed in a 2:1 soil plus sand substrate, contained in plastic trays and moistened with 7% rotation capacity. After sowing, the trays were placed in a cold room at 10 degrees Celsius for 7 days and then transported to the plant grow room at 25 degrees Celsius for another 7 days. Results were expressed as a seedling percentage. In accelerated aging tests, samples with 250 seeds were conditioned on a metallic mesh in gearbox plastic boxes with 40 millimeters of distilled water. These boxes were incubated at 42 degrees Celsius for 48 hours in a BOD chamber. Then the germination test was performed with 4 subsamples of 50 seeds. The evaluation occurred 5 days after sowing. Results were expressed as average percentage of normal seedlings for each treatment. A statistical analysis was performed using Sysvar statistical software. In analysis, when a significant effect of treatment was verified, averages of Scott-Knott tests at 5% of probability were used to test the significance of differences among treated averages. The value of fungal incidence was previously transformed into square root x plus 1. Regardless of the product used for the treatment and of cultivars, seed germination was reduced in the treated and stored, and the smallest seedlings were retained when the seeds' quality after storage were assessed. In this study, when the seeds were stored, there was phytotoxic effect treated by the contact time of the products with the seeds, and when the treatment was done after, there was storage phytotoxicity by the entry of the products within seeds due to the damage caused by storage fungi. There was a significant effect on the 3 cultivars of the storage fungi, Fusarium, Penicillium, and Aspergillus. The cold test is important both to select the lots for sowing and to test the fungicide efficiency in relation to the persistence of the product and the phytotoxicity in the seedlings, since by being in direct contact with the surface of seeds in a high moisture condition, the product can be absorbed by the seed or leached. For seeds from the NS7494 cultivar treated before the quality test, there was a better performance in the vigor of seeds that received the T1 treatment, and was maintained with the T5 treatment by the cold test. It can be verified that when the seeds were sown and left for 7 days at a temperature of 10 degrees Celsius under humidity of 70% of the rotation capacity, there was abrupt entry of treatment products into the seeds, causing a below expected performance for the referred cultivar, probably due to phytotoxicity. In the accelerated aging test, in which high moisture and high temperatures favor fungal proliferation, it was verified that it showed high vigor for treated and stored seeds, and low vigor for the seeds treated before the quality test in any of the cultivars, which confirms that the contact time of fungicides with the seeds is relevant for the complete elimination of the fungi before planting, since pathogens present on the soil or in the seeds reduce the extent of soybean plants. All mixtures containing carbendazim and thyran is in its composition controlled more efficiently the fungus regardless of the treatment time for the NS7494 cultivar. Stored by itself contributed to reduce the percentage of fusarium in seeds from the same cultivar, except in some treatments. In the NS8693 cultivar, all treatment products were efficient in reducing fusarium in relation to control. As for the NS7338 iPro cultivar, the chemical treatment in contact with the seeds for 2 months, so prior to storage, contributed to eliminate the fusarium regardless of the product used. For the NS7338 iPro cultivar, when seeds were stored and treated before or after storage for 2 months, as for the treatment 1, T4 and T5, these products reduced the incidence of the fungus. There was a reduced incidence of penicillium when the seeds from the cultivar NS7494 were treated with T1 and with T4, regardless of the time when the treatment was performed. All treatment products acted to reduce the penicillium when the treatment was performed immediately after harvest in relation to the control. However, the combination that most contributed to the reduction was the product in T1. For NS8693, the treatments containing carbendazim and thyram in its composition were efficient in the control of penicillium, regardless of the treatment time of the seeds. The carbendazim fungicide group act to inhibit the DNA synthesis of fungus, whereas those from the thyram group act to inactivate the essential enzymes. Therefore, these fungicides are extremely efficient in fungus control and seed protection. For any of the 3 cultivars, the T1 mixture was efficient in reducing penicillium regardless of the treatment time. The combination of treatment T4 in contact for 2 months with the seeds from cultivar NS7494 and NS7338 iPro was efficient in reducing the fungus. Additionally, when the seeds were treated soon after harvest, this product did not act to reduce the fungus from the NS7338 iPro cultivar, since there was no difference from the control. For the 3 cultivars, the products in T1, T3, and T5 were efficient to reduce the aspergillus, regardless of the application time of the products. In conclusion, the seed treatment before the quality test was not detrimental to seed quality under optimum conditions. However, under stress conditions, the seed performance was compromised due to the product concentration, except for the use of mixtures containing thiaclopyr insecticide in the composition. The contact time of products with the seed was not sufficient to reduce the incidence of the fungus, Penicillium, Aspergillus, and Fusarium, except by using products based on carbendazim and thyram, which contributed to the reduction of all fungi, regardless whether the seeds were stored or not. When using the products in treatment T1, there was better expression of the physiological quality of treated and stored seeds. Seed treatment products require at least 2 months to be effective in controlling Penicillium and Fusarium. The mixtures containing carbendazim and thyram in its composition are efficient in the control of pathogens regardless of the application time of products. And the storage acts in order to reduce the fusarium and increases the aspergillus. I would like to thank you all for watching this video.